This is episode 111 of the Immunology Podcast, Synthetic Immunology with Dr. Daniel Goodman. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Daniel Goodman from the University of Pennsylvania on the podcast to talk about his research on high-throughput approaches to reprogram immune cell behavior. We've also got our usual roundup of recent highlights of the immunology news coming up, but first...
It's almost time for IUIS. We cannot wait to see you in Vienna. Be sure to plan your meeting and check out the program at iuis2025.org and find some time in there to come visit us at our booth. Also, we'd like to highlight a new resource from the American Association of Immunologists for researchers who may have had their funding terminated or frozen recently. They have launched a Restore grant program that provides up to 1 year of funding for AAI member-led research projects. So you can learn more about this initiative at aai.org/awards/restore-grants-program. Applications close on August 28th, so make sure to check them out soon. Hey, Jason.
Hello.
Happy birthday, belated birthday.
Yes, indeed. I have...
How are you feeling?
Well, good, I suppose. It is my magical year of wisdom. I'm 42.
It's the meaning of everything in the universe.
Yeah, for 1 year, I'm truly wise.
Yeah, do you feel enlightened?
I do. I've been here long enough to know not what to do now.
Are the dolphins talking to you now? Singing to you?
I haven't had a dolphin sing. That would be cool.
Well, congratulations again on another round around the sun. We should celebrate when we see each other in just a few days.
I know. It's just like a week out or so.
I know. How exciting. Well, guys, whoever is going to IOS, you should, come visit us. We'll have a booth. I don't remember the number, but we'll be there, very visible. And we're having, our live show. So everybody's invited to attend. And we're also be recording with attendees. You can come and talk to us, hang out with us in a recording studio. It's going to be very exciting.
Yeah, and you don't even have a time zone change this time, do you?
I know. Finally, it's going to be a very easy transition.
You didn't have one when you went to South Africa either, right?
No, just like an hour or so. That was also pretty good. But Vienna is just like quick 2-hour flight, and it's going to be very convenient. You, on the other hand, have a long way for what we discussed. You're also going to be making some pit stops.
Yeah, I gotta stop by Nestle HQ on the way over. I figured if I'm coming out, I should go to headquarters. Actually, that's where my boss is, so I'm gonna go say hi to them in person and visit headquarters for the first time.
Boo-hoo! You're gonna get shit. You're being sent to Switzerland. I
know, it's so rough.
Sorry, I cannot feel bad for you.
Yep, no, it's not too bad.
Make sure to bring some chocolate. You must bring some chocolate.
Oh yeah. Well, the nice thing is it's the European Nestle chocolate is delish.
Yeah. Well, the Swiss invented milk chocolate, right?
I do.
That's where it comes from.
I do. Yeah. It's awkward being from Pennsylvania though now, because of Hershey's is here.
Oh, Hershey. I'm sorry. Hershey's is not chocolate. I'm sorry.
Well, interestingly, Nestle sold its U.S. brands to Hershey. So Kit Kats in the U.S. are made by Nestle, or made by Hershey, not Nestle.
Oh, I didn't know that.
Yeah.
Well,
my kids love Hershey Park, so it's a whole thing.
Yeah, I should. No.
There's no Nestle theme park. I can't go to Nestle Land. I want to go to a Nestle Land, but I can't.
The whole of Switzerland is Nestle Land. It looks like a Nestle commercial. At least it's not a Milka Land, like the with the purple cows. All right. Enough of this. Let's move to the science, because that's what our listeners have downloaded this episode for, not to hear us drool over future chocolate. Purple. So,
all right.
What you got?
I'm going to do respiratory viruses and cancer.
All right. This
was cool. I love it. So it is titled, coming out July 30th, Respiratory Viral Infections Awaken Metastatic Breast Cancer Cells in Lungs. First author is Shi B. Chia, and last author is James de Gregori. All right, so they did a study where they used a known breast cancer model in mice that spontaneously develops metastatic tumors over time that seed in the lung. But then they said, what happens if you infect the mice with an infection ahead of time? What does that do to it? And the answer is, if you give the mice influenza, and they do all this work with influenza and then come back and say, oh, look, it's true for SARS-CoV-2 as well. But most of the work for the paper is influenza, and then like the last section's like, here's 5 extended figures and 1 main figure where we did it in COVID-2. Long story short, it leads to the loss of the dormancy phenotype in the cancer cells. So it causes these dormant, can't disseminated cells to start proliferating. There's an expansion into lung mets within a couple of weeks, and it's IL-6 dependent. So the inflammation and the infection drives IL-6, and if you get rid of IL-6, it doesn't work. And the way it seems to impair T cell, these cancer cells seem to impair T cell activation, they also ferret out and figure out what, cell mechanisms it is. So the paper starts, hey, we have this phenotype. When you infect them, these dormant cells that take forever start popping up. And so they pop up really quickly. It's pretty bad. Infection drives it. There's a phenotype transition that occurs. There's more proliferation. There's a mesenchymal to epithelial shift. It's not everywhere, but it's significant. They do sequencing and look at all of this. They look at other genes related to angiogenesis. They map a lot of this stuff out. But long story short, there's a shift, but it's not a full shift. If you get rid of IL-6 in an IL-6 knockout mouse, it doesn't happen. And then they're trying to figure out how, what cells are doing this. So, okay, IL-6 is important, but how? And they then identify the CD4 T cells as maintaining these awakened cancer cells. And so they looked and seen that they're in the lung. When you give the infection, there's a CD4 and B cell lymphoid associated tissue, inducible bronchus associated tissue that pops up. And then the CD4 cells seem to be very close by microscopy. So that was their clue. And then they did CD4 depletion, presumably by an antibody. They don't actually say it in the results. And there you go. And it gets rid of the effect. So what looks like is happening, and then they look at reprogramming. They see these CD4 cells are more suppressed, are more of a suppressive or suppressed state after infection. And that's letting these tumors then grow. So long and short, the CD4 cells turn off the CD8 cytotoxicity. And then they look at human data and look at that since the pandemic, it was a natural experiment, and they do see the cancer incidence go up.
It's because you have an overactivation of CD4 cells? No. Or is it the other way around? Sorry, I think I missed that. So the ultimate consequence is that you're inhibiting CD8 T cell activation and cytotoxicity towards the cancer cells, right? And that is because the infection triggers the suppression. How is that connection again?
So infection triggers IL-6,
Yeah.
Generally. That leads to CD4 remodeling into a suppressive state, which turns off CD8 cytotoxic T cells, and so the tumors can progress.
And because usually IL-6 is kind of associated with the opposite, with like pro-inflammation and
so
damage.
Right, the inflammation and the lung damage, right, then has so you have inflammation. There is in the lung from an infection, which is where the tumor cells are seated. They're in the lungs already, quiet. And then what happens is then to turn off the inflammation over time, you have more CD4 cells that wants to control things. So that sets up
Mm,
suppressive states. You don't want CD8 cells run amok in your lungs after an
yeah,
infection. And so turning
right.
off that then allows pop goes the weasel. Because
Yeah,
it's not like they get this right after they're infected. This is weeks after the
yeah.
infection.
As part of the normal recovery to, I guess to protect the tissue, to the normal reduction of the inflammation or recovery from the inflammation from the infection, which is something you have to do because you kind of need your lungs to be in good condition even after a viral infection.
Exactly right. So it's like trying to hold things in check. By holding things in checks, cancer comes back.
Oh, okay. Damned if you do and damned if you don't.
Yep.
It's interesting to see, the impact of there's so many diseases that get triggered by an infectious episode, and it's interesting to see different mechanisms by which it's not it's having an infection is even if you recover from the infection itself, it's not free. You're going to have these delayed, some examples, if you have autoimmunity can develop, or now you're having uh, kind of a dampened immune response that allows cancer to get activated. Yeah, it's not a good idea to get infected if you can prevent it, I guess.
Nope.
All right. Okay, so for my first, I have two really cool stories. I want to start um, with the first paper, which is titled Determinants of Successful AAV Vector Delivery of HIV-1 HIV-1 broadly neutralizing antibodies in early life. And this was published 30th of July. First author Amir Ardeshir from corresponding authors Mauricio Martins. And this is at the University of California Primate Research Center. And I think the University of Florida. So Scripps Institute at the University of Florida in the city of Jupiter, Florida. I always find that funny name. Um, I think it's a really cool concept. I'm not sure how in the end applicable it is, but I think it's a nice way of looking at potential therapies for preventing HIV infection. And this is a study done on macaques, mouse models, rhesus macaques, and which to study HIV, I think they're kind of the best thing available. And the study, is circles around the idea that if you can have, broadly neutralizing antibodies, which are these antibodies that are really good at neutralizing all kinds of kind of HIV variants, and they can provide kind of durable immunity and can prevent the infection by an HIV particle. And so they're really associated with protection against HIV and the development of AIDS. And so one of the ways, of course, so this that if you could have in your blood broadly neutralizing antibodies in sufficient titers, that would kind of protect you against HIV infection. And so this is one of kind of a strategy could be to infuse people with broadly neutralizing antibodies, and especially infants, which are which they are. I guess everybody doesn't want to have HIV, but I think infants are probably the worst case scenario. But of course, that's really hard. If it's already hard enough to get retroviral drugs to people that need them, imagine having to infuse them with broadly neutralizing antibodies. But then, so what they suggest here is, what if you could develop a one-shot solution that allows for the broadly neutralizing antibodies to be produced by the person themselves, and this is by basically transferring the genes for the production of the broadly neutralizing antibodies using an adeno-associated viral vector. And so what they do in this study is that they have, this, they do these studies in rhesus macaques, and basically they treat newborn monkeys because it's important. So one of the issues with broadly neutralizing antibodies is you can generate immune responses against the broadly neutralizing antibodies, and then you lose their functionality. But infants are known to be less prone to develop this kind of anti-drug antibodies. And so what they do is they have this macaques, and they basically they give intramuscular injection after birth, I think within the first 48 hours, of these AAV vectors that are expressing this particular broad neutralizing antibody called 3BNC11 117. And they show that by doing this, they can actually induce production of this antibody in these monkeys after some time. I think it's 2 weeks, 1 or 2 weeks after the injection of the AAV. And what they show is that this actually can protect these infant macaques against acquisition of HIV infection. In this case, they're using the simian HIV virus, and they show that that the production of these antibodies correlates with protection from infection. And so they kind of want to understand, no, okay, this seems to work. What are the conditions? How, if we were to administer to infants, what are the constrictions regarding time? And they show that, well, they try to understand how far after birth you can do this treatment and still have protection. And so they test, they show that if you inject these AAVs into more juvenile and adult rhesus macaques, they will most likely mount anti-drug antibodies, which will render the production of the broadly neutralizing antibodies kind of impossible. And so they really show that as long as you can kind of stick to the first, I think 4 weeks, 4 weeks after birth, that is kind of the sweet spot to introduce in these broadly neutralizing antibodies. And they follow the monkeys in their life, and they show that they are still expressing these antibodies later on in their life. So they can kind of, this protection is extended to kind of adolescence, which in a human situation it would be very good, because then you can protect also from transmission, sexual transmission, for example. And so they do a lot of studies looking into understanding the expression of these vectors. But I think what's interesting is that they also test an idea of trying to make sure that to reduce the chances of antibodies against this broadly neutralizing antibodies. They're also testing if they expose the monkeys in utero to the neutralizing antibodies with the mothers. So if they, for example, transfer this antibody during late pregnancy to the mother, and which then gets transferred through the placenta to the fetus inside, then this can actually increase the chances of tolerance, and then reduce the chances that even if you are injecting the AAV therapy a little bit later in life, then there's a reduced chances of generating antibodies. So that kind of improving through fetal exposure to these recombinant antibodies can tolerize the monkeys, and inhibiting the production of antibodies against this drug, and improving the expression of the broadly neutralizing antibodies, even if you are delivering it later in life. So I thought, in principle, that was a study. So they follow the monkeys in time. They show that this as I mentioned, this protection can be long lasting. And I think in general, they make a really good point on this could be kind of a one step therapy that you can do at the moment in which most in the places where um, this therapy is needed. That's usually when you have the most chance of having access to the baby and the mother is right after birth. That is the moment in which you're most likely to be able to have access to the child, because then later one of the issues with, for example, retroviral drugs is that you need constant follow up, and that gets very difficult, especially in places where the infrastructure is missing or people have a hard time physically moving to the places where they have to do the follow-up studies. And yeah, and transmission through, for example, breast milk, or it's a problem in babies that are breastfeeding, and they could be protected from this using this therapy, for example. So I thought it was very interesting. I think application will be probably pretty challenging, but I think that as an idea, I thought it was pretty interesting, a pretty um, interesting approach, and maybe an alternative. And they also suggest that you can think about this for other diseases that are also, like, for example, they mentioned malaria. There are also, I think, broadly neutralizing antibodies for malaria known. So maybe you could also think about this for other kind of drugs that are really hard to treat, and vaccination so far has not shown enough success.
It's interesting. I think an adenoviral vector at birth has a huge set of like, ooh, do we want to do that? But,
Yeah,
we're doing this in monkeys to understand science right now, which is different, right? But I think
yeah,
super interesting.
yeah. I mean, yeah, all these things is very aspirational, I would say. But well, you understand. It helps you understand, and you never know. Maybe something changes in the future, and we can still
And that's
do
fine. I'm okay.
something around this.
All right. Well, I have no segue here for HIV, but I am still on the tumor kick.
Mm.
And
All right.
in Nature Metabolism, published the 25th of July, the source of dietary fat influences anti-tumor immunity in obese mice. First author, who I'm going to butcher their name, I am sorry, is Berta Kunka-Moller. And last author, this is from the lab of Lydia Lynch. All right. So we know that there's an obesity problem in the world, especially in America, but in the world is getting a little more rotund. And we know that it affects your immune function and a bunch of other stuff, right? It's not just the straight effects of the cardiovascular metabolism. Being obese can have knock-on effects. So they did a study where the same total calories went in, but one set of mice got animal fat and another set of mice got palm fat. There's this whole thing about, is palm oil or beef tallow, beef lard worse for you? What's worse? And the answer is, for this cancer, the beef is worse.
So I just want to say beef lard would be the fat from the beef, right? Because lard,
Correct.
I don't think, is such a common word for non-English speakers.
Yes, it is the beef fatty, the tallow. So they did a bunch of different fats to begin with and found the biggest split between palm oil and lard. They did butter, excuse me. They looked at lard, beef tallow, butter, coconut oil, olive oil, or palm oil, and selected the two ends, which were butter, fat, and palm oil being the two most variable from each other. So they fed the mice for 10 weeks, then they did the B16F10 melanoma model and found that the lard-based mice had more than a standard fat, and the tumor growth dynamics in the high-fat diet that's fed mice were significantly different based on the dietary fiber. So basically anything animal-derived was bad, but then they focused on butter-based and palm oil, which was a high-fat control versus standard fat, right? So now you have a three-arm approach of high fat generally, but then bad high fat and not. And the bad high fat often made things worse. The bad high fat was always a worse state. So standard fat on one end, good. Bad high fat on the other. This plant fat in the middle. It's generally how it goes. So they looked at syngenic tumor models, including the E0771 breast cancer model, the LSL KRAS lung model, pancreatic ductal carcinoma, melanoma, and colorectal. Some grew faster, not all of them did. So the breast cancer one did, and that LGSP tumors grew, but there was no difference in the pancreatic or YUM tumors. And the subcutaneous MC38 tumors also grew faster, but the palm oil was not significantly different from either. So this is like, okay, it depends on the tumor as well. And they're trying to figure out what the heck's going on here. So global metabolism was not changed between them, but they saw differences in infiltrating immune cells. And so they looked at flow and found that there were changes there was less infiltration of NK cells, CD8 cells, and FOXP3 positive CD4 cells into the tumors in the high fat animals. Okay. And then, but there was more NK cells in palm oil than butter. So NK cells that was splitting out as a signal of like high fat bad, but something's extra bad in animal for NK cells. So they looked at that as one thing. And then they looked at cytokine production, and there was less interferon gamma in the tumors of the high fat mice, particularly the butter high fat. And then they looked at the metabolism, and they found that NK cells had jacked up metabolism and function. So they took the spleens from these mice, and they saw decreased interferon gamma. There was neutral lipid staining accumulation in them, but there was a lot less of that in the high fat palm oil group, and clearly a lot less in the standard fat group. And then they had decreased metabolic function, oxidative burst in the NK cells, and they just looked damage, right? And they looked at mitochondrial proteins and saw damage there. And then they looked at a whole bunch of genes, because who doesn't do proteomic data? So a whole bunch of the proteome to identify different genes that could be involved in here, and a whole bunch of them were showed to be downregulated that were important for NK function. STAT5 trended that way in palm oil. Just there's a bunch of things. It was better in palm oil than high fat, but not quite significant. But long and short, a lot of the NF-kappa B pathways and a lot of just genes that are associated with NK cell activation and cytotoxic function were all downshifted, particularly in the butter high fat. And then they identified through CHIP X enrichment analysis that this is a lot of this was due to CMYC. And then they were like, well, what is the protein? They looked at all the different lipids. So they compared the lipid composition of a butter high fat and palm oil high fat, because there are some differences in saturated fats and unsaturated fats, even though it has the same calories. They looked at C18, that didn't have an effect on there, but they eventually then looked at the metabolome and tried to figure out what was differentially expressed in the metabolites. And sterile carnitine, CAR180, seemed to be a culprit of something different, and they went down that rabbit hole. And it's not difference in oxidative rate metabolism, but the C14 and C18 carnitine ones, the levels were based on the fact there's more input, right? So more in the butter leads to more change out. They're not converting it more or less. There's just more of that type of precursor, the C18. And so you get more on the way out. Standard Le Chatelier principle. And they found that CAR18 inhibits CD8 T cell mitochondria and function. And so you see a kind of multiple effects where you have NK cells just having too much fat in them and them stop functioning, inhibition of the CD8 cells. And they think between those two, that is why this diet in particular makes the immune cells cry and the tumors get.
I thought that, but so you're telling me there's more C18. So C18 is palmitate, so palmitic acid.
Right, which is,
Right?
but it's the fully saturated version, right?
Yeah.
No, it's C18 zero, so there's no double bonds. And so animal fats have more saturated fat than palm oil, which has more mono and polyunsaturated.
Oh, okay. Yeah. Yeah.
So like there's C18.2 and C18.4 and all that. And so there's more.
Right. And also butter, because I thought butter, I thought it was made of butyric acid, but I guess it's only a bit of it. That's where the word butyric acid comes from. C4.
Yeah.
C4? Yeah. Um, but I guess it's only a little part of what. So the main component of butter is C18.0.
What's one of the major ones?
Mm.
And that
Yeah.
one, being such a major component, pushes more of this palmitoylated or carnulated version just by
Yeah.
more in equals more out. And that's
Yeah.
bad for the CD8 cells. But just all this animal fat makes the NK cells a little stuffed, and they don't like it and don't function as well with a big old stuffed cytoplasm.
Yeah.
Their mitochondria are crying out in distress.
Indeed. Indeed. So take care which fats you're taking, especially if you have
But there's a
cancer.
whole thing that palm oil is supposed to be really bad for you, remember?
Yeah.
The rage right now, we're supposed to be eating more animal and so which is it? Which is it, science?
Well, but I think also palm oil, also hydrogenated palm oil is also bad. I don't know.
This was
So
just
I,
natural. This wasn't,
mm.
hydrogenated on the
But I
other side.
think palm oil is not that palm oil is bad for you, it's that palm oil is bad for the environment. Making palm oil is extremely,
Yeah, there's that, but it's also supposed to be worse for you. And like there's a whole movement against palm oil and pushing for animal fats. But this is part
oh,
of, the new political push that we're experiencing
yeah.
for some of
Yeah, I
us.
don't think that's necessarily based on evidence. I think it's just
It's
based on
not
feeling.
necessarily, but it's very interesting. This paper is now out.
So you're telling me that what's his name? A carnivore diet is not good?
I don't know. I'm not, yeah.
What's his name? Choose, male
There's the Liver
influencer.
King, right?
Uh, yeah. All right. So basically keep your fat content low if you want to NK cells to take care of that cancer for you. Makes
Yeah.
sense. All right. So closing up, I also don't think I have a good segue for this, but I have a really cool study. And this is one of those studies that you think this is either huge or it's going to blow up. I don't know how to interpret this because, yeah, if you take it face value, it's quite extraordinary results. And I really wonder how much of this you're actually going to be using in the future. So, without further ado, the article is called Lithium Deficiency and the Onset of Alzheimer's Disease, was published in Nature August 6th, so it's fresh off the press. First author, Livio Aaron, from the corresponding author, Bruce Yankner, sorry, at Harvard Medical School. And yeah, I just think it's quite extraordinary because it's such a simple, like the story is so simple that it's almost too good to be true. Basically, in this study, they find that the deficiency of lithium can be a major factor in the pathology that we see in Alzheimer's disease, and that part of this pathology could be reverted by providing an exogenous lithium to increase lithium concentrations in the brain, and that this can, at least in the mouse models that they test, improve cognitive function and reduce kind of cognitive decline. So let's just go step by step. What they do in this paper is that they are looking, so they start testing samples from patients, I guess postmortem, from brain of patients that have, that die with different levels of cognitive impairment or, control patients, also patients that have died from Alzheimer's. And they quantify metal levels in the prefrontal cortex, which is one of the predominantly affected regions in Alzheimer's, cerebellum, which is relatively unaffected as a kind of a control. And of all the metals they look at, one showed the highest significant difference, which is lithium. And they see that there's reduced levels in the prefrontal cortex of individuals that have both Alzheimer's disease and uh, cognitive impairment. And they kind of look further into this, and they show that in patients with Alzheimer's disease that had deposits of amyloid plaque aggregation, so alpha amyloid beta aggregation, they showed that there is some kind of capture of lithium ions inside these aggregates. So they have like this laser laser focused this dissection of the samples, and they can really look into the plaque, the areas that have high plaque formation, and they have a increased concentration of lithium in the amyloid plaques. So they kind of start, kind of looking further into this, and they look into mouse models of Alzheimer's disease, and they again show that that there is this lithium sequestration by alpha amyloid plaque deposits in the brain of these mice that develop this deposition of amyloid plaques. So what they do is that if they feed mice, so they have control mice, and they also have mice that have models for Alzheimer's disease that develop plaque formation, and they remove lithium from their diet, what they see is they see an increased accumulation of amyloid plaque deposits. And also in some models that develop phosphorylated tau aggregates, they also see an increase. This is this G20AD mouse model. And basically, they have increased Alzheimer's disease-like pathology when lithium is removed from their diet. And they look into they basically see increase also phosphorylation of tau protein in deposits in these mice. And this results also in impaired cognitive function, but also in wild type mice. Not only mice that are developing Alzheimer's disease, but also in aged wild type mice also showed significant memory loss and certain cognitive impairments that are kind of similar to what people see in Alzheimer's disease. And so they look into what is going on in this mouse brains, and they look at all kind of, they see a lot of differentially expressed genes in which basically they see changes in brain function at the molecular level. And one of the things, because this is the Immunology Podcast, they look into microglial function, and they see that when it comes to the resident microglia in this mice, they see a reduction in the number of microglia that express the gene CXCR1, which is this homeostatic marker for microglia in the brain. They have increased expression of ApoE, which is associated with increase with Alzheimer's disease development. Basically, they have a more inflammatory microglial transcriptome. And this also associates with kind of other markers of reduced neuronal function, astrocytes. So everything is kind of affected. There's a very widespread changes in transcriptome of these cells in these brains. And so what they show is that this is due to the removal of lithium from the diet. And they trying to understand what is lithium doing, how is lithium, why is lithium being picked up and affecting the cells. They zero down on the Wnt-catenin signaling, because they show that lithium seems to, in a way, the efficiency of lithium elevates the levels of this kinase GSK3 beta, which phosphorylates beta-catenin, and therefore degrades it, that targets it for proteasomal degradation. Also interestingly, GSK3 beta can phosphorylate tau, which is also phosphorylated tau is a feature of Alzheimer's disease. And so increased tau phosphorylation is observed upon lithium depletion. And they think that this is, in a way, probably one of the mechanisms by which lithium availability is affecting brain function. And they show that they reduce some of these effects by inhibiting GSK3 beta with a inhibitor, CARE 91021. And this can, to some extent, counteract the effects of reduced lithium availability. And so what they say, well, if the problem is that there's too little lithium, what happens if we give mice a bit more lithium? And the idea is that by increasing the bioavailability of lithium and reducing this inflammation, and also because lithium tends to be taken, kind of captured by these amyloid plaques. So is there some kind of lithium that reduces the chances of being kind of sequestered and not bioavailable anymore? So they test different. So they suggest that maybe the electrostatic interaction of the lithium ion, which is positively charged, with the amyloid deposits might be kind of affecting the level of sequestration. So they test different ionizations from different types of lithium salts. So we all know, I think lithium carbonate is most commonly known lithium salt, the one that is, the one that is used for bipolar disorder, I think is most commonly used clinically. But there are other, lithium can make salts of many types, including organic salts. And this is where lithium orotate comes into play, which is a lithium salt that has a carbonated structure, a organic carbon structure. And they show that this salt has lower conductance, so the ion is less likely to separate from the whole salt. And so they compare treating mice with either lithium carbonate, or lithium orotate. And basically they show that lithium orotate has the ability to be less, so make more bioavailable lithium in the brain that is less sequestered by amyloid plaques, and that this can actually ends up being very effective at reducing the alpha amyloid deposition, phospho-tau accumulation in the brains of these mice, and basically by just adding it to the diet of the mice. And this has effects on kind of cognitive decline, reduces cognitive decline, not only in Alzheimer mice, but also in old mice. So that seems pretty interesting. And they measured this. They show that also in old mice, they can reduce age-related production of pro-inflammatory cytokines in the brains, IL-6, IL-1 beta, microglia activation, increased degradation by microglia of amyloid plaques, which is kind of one of the jobs of these cells that they're not doing that well in Alzheimer's. And in principle, just they show that this seems to correlate with improved cognitive function in these mice. So basically, kind of long story short, the authors suggest that at least part of the pathology of Alzheimer's disease is caused because of a reduction in bioavailable lithium in the brain, that it gets captured by amyloid plaques, that this can be reverted by increasing by supplementing lithium using salts that are less likely to be sequestered by amyloid plaques, and that this can improve cognitive function not only in Alzheimer's, in the Alzheimer's disease, but also in aged mice, in aged brains. So very interesting. I wonder where this will go.
I mean, that's very interesting. I don't know. Like mouse dementia and human dementia aren't the same. That all the drugs have failed probably because of that.
Yeah. So they mention, and I just was looking a bit, they mentioned that there are some studies in which availability, like consumption of lithium, kind of epidemiologically, has correlated with lower dementia incidence. There's some study in Denmark, and there's also studies on people that take lithium for bipolar disorder. So there seems to be some data in humans.
That's controlled against people with bipolar who don't take lithium.
Yeah, I don't know. I didn't read the actual. I
Right, like
didn't read
that's
the
the
reference.
question, right? Like,
Yeah,
is it bipolar disorder or lithium?
yeah. Uh, I'm not. I don't know. I cannot answer that question now. But yeah. So I think looking at the references and kind of preparing for this, there seems to be already um, some literature suggesting that lithium can help for um, yeah, mood disorders, behavioral disorders, but also
Oh, sure.
for aging. And but as you mentioned, we were discussing before, like the problem is like uh, lithium carbonate is a bit heavy on the kidneys, so it's not like there's a disadvantage. There's a potential detrimental effect of taking this as a supplement just for your brain function. Hypothetically, they do say in this paper that this organic salt, is less associated. They did not see any toxicity in the mice. So they suggest that maybe this or lithium orotate could be have less side effects, so be safer to test.
Maybe. I mean, it'll be interesting to see.
Indeed.
All right. Well, we are going to be speaking with Dr. Daniel Goodman at my old home location of the University of Pennsylvania in just a moment. But before we get to that, Brenda, you do immunotherapy research, right?
Indeed.
Well, you know
That's
what?
something for me.
STEMCELL Technologies offers products and protocols for immunotherapy research, including T-cell isolation, activation, and expansion reagents. Use EasySep T-cell isolation kits to isolate highly purified T-cells in as little as 8 minutes. That's more time back to do more experiments, Brenda.
Woohoo!
Follow up with Immunoccult reagents designed for human T-cell activation and expansion. Learn more about STEMCELL's optimized protocols and reagents for immunotherapy research at www.stemcell.com/T-cell-therapy.
Now, for the second part of our show, we're so happy to have our interview guest. We have with us Dr. Daniel Goodman. He is assistant professor at the Perelman School of Medicine at the University of Pennsylvania. And I'm very excited about this conversation because one of the topics of his lab is synthetic biology and genetics in T cells at the moment. And I just find those topics so cool. So I think we're going to have a really nice conversation. Dr. Goodman, thank you for joining us at the Immunology Podcast.
Yeah, thanks for that introduction. I'm really excited to be here and talk to you.
Thank you for being here. All right, Brenda, we'll start with T cells and you. So go first. I'll hold back for the second part.
Yeah, I know. Yeah, exactly. You can talk about that, and I'm going to talk about my favorite cell type, which is the T cell. Everybody knows is the superior cell. Now, it's a very interesting cell type. And I think part of the work of your lab, which you recently started at University of Pennsylvania, congratulations, is working on synthetic biology. And I think because in a way, T cells are such good sports, I think, for genetic engineering, and you can do so many things with adding genes to T cells, and they're so amenable for genetic engineering that you can do so many cool things. So I would say maybe why don't you introduce to us a bit about what is the work that your lab is doing around synthetic biology in T cells, and kind of the previous you've been working on screening for CAR T cell development. So what excites you the most, and what is your lab working on when it comes to engineering T cells?
Sure. Yeah. So I'll start a bit about my background and how I got to where I am, just very briefly. As it was alluded to at the very beginning, my training really started in sort of more theoretical bioinformatics, computational biology, and then I moved into bacterial genomics and genome engineering. I did my PhD with George Church at Harvard and MIT and the Broad. And there I really fell in love with a lot of the new technologies that we had at the time, and they're still, pretty exciting developments there. Sort of before CRISPR, even being able to synthesize DNA at scale and test lots of genes and genetic elements and protein variants in these big pooled screens and measure the function of genes and proteins at scale. And then you get these beautiful, very high diversity, high complexity data sets that you can then use to learn how DNA turns into the function of proteins and cells, and how you can engineer with that kind of data. And then, I love E. coli. I love working in bacteria, and it's very fascinating, a lot you can do with them. But I was always been fascinated by the immune system. And especially at that time, that was the very birth of CAR T, which I guess we'll talk a bit about what probably most of your listeners don't know about CAR T, but we can talk a bit about it and sort of the next steps in that of engineering T cells beyond CAR T even. But I saw that happening, and I saw, as you alluded to, how powerful T cells can be and how amenable they are to these approaches, and the ability to scale and expand these cells, take them out of the body easily. And so I thought, what a great opportunity to apply the tools that I developed and learned about in George's lab to the immune system, and in particular to T cells. So then I did a postdoc with Alex Marson and Cole Roybal at UCSF, where we built a lot of the technology allows us to do these things at scale and do pooled screens on different CARs and genetic variants, and manipulate the genome of T cells at scale, try thousands and even hundreds of thousands of different genetic variants, and test them all in these rapid, high throughput multiplexed approaches that I had sort of pioneered in E. coli in my PhD. And then that worked very well. It took a lot of technical expertise and a lot of time to get those things working in mammalian cells, but we're there now. And some of those things have borne fruit. We have some papers from my postdoc, and then some of the work is just kind of come out in the coming months here, scaling quite a bit beyond what we've done before. And then my lab, getting to your initial question, is really going to take that approach further and try to think of all that we can bring to bear, all these new technological tools, CRISPR and scaling the editing of genomes with human viruses like AAV and lentivirus, using non-viral approaches in LNPs, barcoding individual T cell clones and tracking them in animal models, understanding how to manipulate the sort of natural circuitry of T cells for engineering goals, with really the ultimate goal. I'm at UPenn, where, of course, I'm at, in addition to the School of Medicine, I'm at this place called the Center for Cellular Immunotherapies, which is where Carl June's lab is, and a lot of famous labs, Sarah Gill, Dan Powell, a lot of labs focused on deploying, immune cell engineering for therapeutics, including CAR T and other things. So I think it's a great opportunity to take these engineering approaches we've developed and really put them into products that can get to patients and help primarily with cancer, but increasingly we're looking at other diseases like autoimmune diseases as well.
Can you elaborate a little bit on what you mean by engineering?
Yeah, that's a great question.
How are you engineering it? And, how is it engineering and not just, for instance, I cause random mutations all over the place and try to screen to figure out what happens, which is not engineering. Yeah,
I completely agree. So, my undergrad degree and my PhD are both in bioengineering. I think, that it's a bit of a philosophical question, but I would say we absolutely do not do random mutation. I think that's, that's very powerful in some cases, depending on the scale of your approach and the question you're trying to ask. But we really try to target our modifications. And I guess there's always this trade-off between different aspects of scale. So you can look at a single protein or a single domain or even a single, genetic motif or binding site, and you can sort of manipulate that. Or you can look at many proteins or a whole genetic locus or a whole genome. And as you zoom out the amount of DNA you're trying to understand, the amount of sequence that you're looking at is larger. And so the amount of points that you can manipulate becomes less, right? So I think there's always a trade-off there. We tend to look at smaller things and do larger engineering approaches. So one thing that we're really fascinated by is how T cell signaling works at the membrane. And so there, CARs are kind of fascinating because they take these different modular. It turns out they're quite modular components of membrane signaling in T cells. The CD8 or 4-MBB, or sorry, CD28 or 4-MBB co-stimulation, and then pieces of the TCR. You can put them together into a single protein, and they work quite well. But then there's a lot of sort of engineering scale questions about, well, why does that work, and how can we tune it, and what are sort of the input and output relationships? And we can explore that not by randomly mutating the CAR, but by bringing in other domains, by mutating individual motifs, and really going into the molecular biology and trying to build what I often talk about as a sort of sequence to phenotype landscape of, as you manipulate a protein intentionally in different ways, what happens to the sort of function of that protein. And because we're using synthetic DNA libraries, which I haven't really talked about, we can maybe dig into if you'd like, where we're not just randomly mutating DNA, but we're actually on the computer designing, hundreds, thousands, even hundreds of thousands of different intentionally chosen sequences and testing them all at once. And then each of those sequences then gives us some sort of, functional readout. And then we can sort of put all that data together, and that helps us sort of, understand the engineering problem of what protein sequence we should choose.
Yeah, so I guess the example that would be for what you're talking is your paper at Science Translational Medicine, for example, which you're comparing different CAR T domains. And I see, talking about this approach, so which is something that also we've covered a few other papers from, for example, Alexander Marston's lab. So this idea of testing a lot, and something that you could not do before the age of being able to just order all these gene blocks or these synthetic DNA, make these huge libraries.
Or CRISPR guides, exactly. Yeah.
And there's a lot of kind of smart design around these experiments. Really, what is your readout? What is the how are you going to account for which kind of controls you're going to do? Because I can imagine sometimes when you have when measuring so many options at the same time, things like noise, making sure that your signal is going to be above your noise, making sure that you're not going to be confusing your measurements. I guess those are a lot of really important considerations that I think show from a lot of the screening papers and research projects that we've seen. Would you like to maybe kind of explore the implications around doing these very complex, large-scale experiments? Maybe you can use your CAR T-cell project as an example of how you guys looked at it. If any of our listeners are thinking, I want to do a screen, I want to also take advantage of these currently available technologies, what are the important things to take into account?
Yeah, there are quite a few. I think it depends on a lot of factors. One thing, especially going from a system like E. coli, where it's very easy to get many hundreds of millions or billions of cells, you're really limited with mammalian cells and primary cells, especially. T cells are very nice because of all of the primary cells, and certainly immune cells. It's not crazy to think of 100 million T cells in a library, or even a billion potentially, and so that gives you a level of scale that is pretty unique and exciting. However, T cells do have a big difference from E. coli, or even a cell line. Like a lot of these screens, even mammalian cells are done in 293T or some sort of like standardized cell line. These are primary cells, so every human donor is going to be different, and even T cells from the same donor, obviously there's CD4, there's CD8, and there's so many different subsets of T cells, and some are naive and some are memory. And so when you're putting a library of things, whether it's a gene knockout library, something very simple, or you're engineering a CAR, a receptor, or something else, you have to take into account that heterogeneity, and it often confounds your signal. So the coverage that you need, we talk about coverage in these screens, where how many cells need to have the exact same thing. So you're measuring not just one cell per construct, but hundreds, thousands. The coverage tends to be much higher. It needs to be much higher. And you often need to do many donors, and you need to be very thoughtful about the time course and the scale and all those sorts of considerations.
So in terms of this engineering, you're predicting stuff, right? You're trying to make changes and know what's going to happen and see if you're right. You're also using machine learning, it looks like, just from reading on what you're up to for that. So how is that working, and has it been successful
we're very excited about it. So I was a bit skeptical. I think it started out just like a fun idea because so the reason I have some background in computer science, machine learning. I'm not at this point. The field has gone so far since I was heavy into it that it's pretty incredible. It's hard to keep up. But at first, it made sense simply from the standpoint of if you think about most of these really powerful machine learning models and other spheres like large language models like ChatGPT, they work so well because you have so much data. You can take the whole internet and feed in the internet. Um, and there are protein language models now like ESM and AlphaFold that can take all of the protein sequences or structures and learn from that. But because of the scale of our experiments, it's a great opportunity to sort of ask, can we fine tune a model with all of our data and get it to learn things and understand aspects of our problem of our data that are not clear to us, and maybe it can come up with new ideas and new things to try. And it turns out that that's working quite well. So to give you a brief example, this is all unpublished, and we're kind of working on it now. We have the paper that you mentioned, the paper in Science Translational Medicine. We tested 40 natural different signaling domains in a car, and we found a new one from B cells that seems to work quite well in T cells, surprisingly. But what we really wanted to do was, from an engineering standpoint, right, is understand, well, what is it about that one signaling domain? We know a bit about the molecular biology of these domains. They tend to consist of short motifs that get phosphorylated and bind downstream signaling proteins. So we wanted to go in at the sequence level and the motif level and sort of understand the engineering rules for how to build those signaling domains and sort of rewire signaling at scale. And so we built a library of about 40,000 different sequences, put them into cars and tested them in various ways. And a lot of them were fascinating. Very few were really as good as or better than the classic 4-1BB or CD28 signaling domains you'd use in a car. But we fed all that data into one of these language models. We fine-tuned it and asked the model, first of all, to say, can you help us understand why some of these domains work or don't work? And it does. It's able to pull out the known, from literature, known motifs, like things that bind PI3 kinase and TRAFs and things that are known to be involved in T cell signaling. And it'll say, if you break this motif, then the car shouldn't work. And it's right. It doesn't work. It's because you can do in silico mutational scanning within the model, it can actually tell you what's going on. But then what's more exciting is then we can say, okay, now via a variety of techniques, including diffusion and genetic algorithms, can the model generate new sequences that it predicts to be better that we wouldn't have been able to come up with ourselves. And again, because we can scale these techniques, we can ask it to make not just one or two, but 1,000 of them, and say, okay, give me your 1,000 best guesses about what makes a good car as far as proliferation, signaling, killing, secretion of cytokines. And we've tested them now, and we're just looking at that data. It's just coming back. It's going to go into the paper, and I'm very, very heartened. Many of them are working quite well. We've tested a few in vivo and in vitro, and it's not ready for publication yet. But to answer your question, yes, it's absolutely working, and these models are coming up with some sort of understanding of the rules of signaling in the context of a car, and can generate new sequences that are functional.
That's so cool. So if you can share with us, which are the measurements, which are the readout that you include to train this model?
Yeah. So the easiest measurements are the ones that you can select for. So proliferation is a super easy one, right? But oftentimes there are trade-offs between proliferation and killing. So there are T cells that can proliferate, but then they actually don't become cytotoxic effectors. Or the CD4s proliferate and they don't kill, and the CD8s don't, which is fascinating to learn. But you need many different measurements. So proliferation is one, and we did this in the paper in Science Translational Medicine as well. We also looked at which T cells could secrete cytokines. So you can do intracellular staining, and then fix the cells and stain for interferon gamma and IL-2, and then sort them, and then sequence the DNA barcodes corresponding to those cells and see, aha, these CAR designs are making more IL-2 or more interferon gamma. You can also look at so essentially the nice thing about these techniques based on sorting is that immunologists love to use flow cytometry, love to stain things and look at them. So you can just turn those into sort-based pooled screening assays. You can sort for memory markers, exhaustion markers, whatever you care about. The big problem, though, is that all these assays are traditionally done in vitro. And there's like a dirty secret among these big projects, these big screens that Alex does and lots of people do and we do, where oftentimes when you just measure killing in a dish with tumor cell lines, it looks nothing like what happens in a mouse, and let alone in a human. And so there's this big push right now, and the papers are starting to come out to start doing these screens in mice, to do either in xenograft systems with human cancers and human T cells, or even in syngeneic systems with mouse T cells, so you can recapitulate the real tumor environment, to start screening in the mouse and take the T cells out of the tumor and see which T cells got there, which ones are making cytokine, which ones are killing, et cetera. And so we're starting to do that as well.
So when you go from in vitro is wonderful, right? I can get lots of data really quick. How do you do, but you're talking about how you need large volume for any, AI or any of this engineering, you're talking about high N, right? Yep. So how are you doing high N in vivo?
Well, it's similar, right? So people, my old boss, Jeff Bluestone, would talk about this all the time. The mouse is a furry test tube, right? Especially these NSG mice where, all they're doing is, supporting the cells that are growing. So you can take a library of, 10,000 constructs and you can grow that up into 50 million cells. And then you can give, 10 mice, 5 million cells, and then you take out the tumors and spleens of all 10 mice, and then you sequence them. And then you have essentially 10 replicates of your library, and you can see across those 10 mice which T cells are expanded, which ones got to the tumor, which ones survive, which ones are not exhausted. Maybe you can even do ex vivo measurements on the T cells that come out of the tumor. And that's a high enough N. I think it's certainly limiting compared to in vitro. So in vitro, you could do gigantic flasks, many flasks. You could easily do hundreds of millions of cells. That's much harder in mice and much more expensive. But we actually just finished doing an 80 mouse screen to give you an idea of that scale. And it's certainly a lot of work and a lot of hands, a lot of time, and a lot of money. But it's definitely doable, and we got great data out of that screen.
So I would say, understanding the effect of these genes or looking into these readouts in T cells is all well and good. I mean, it's fascinating as a fellow T cell immunologist. I'm always interested. But oftentimes this is kind of with the intention of improving therapies, and I think particular CAR T cell therapies, which you are pretty much in one of the centers of the world when it comes to of CAR T cell therapy and other cell therapies. So from your vantage point, from your, you also come from, well, I think you've always been in places that have been in the center of this. So I think you're a very good person to ask. Where are we going, right, in terms of where are the challenges? And I mean, we discussed a bit about some of them, and your research is trying to address. But right now, where are the next steps or the next things to look into when it comes to cell therapy, T cell therapy?
Sure. So I mean, there are a lot of obvious questions which you could ask anyone in the field, and they probably give you similar answers. And I'll briefly go through them. I think the most obvious thing is getting these things to work better in solid tumors, and that's a combination of coming up with better ways of recognizing the tumor, or reducing off-target toxicity, and avoiding the sort of suppressive microenvironment that many of these solid tumors create. I think there's a ton of approaches to solving those problems. Screening can work with many of them. We are trying to solve them with screening different genetic modifications to T cells and different types of CARs and signaling. I think understanding how the natural immune system is involved, and why things are getting suppressed, and how you get things like epitope spreading, and how other T cells are coming in after the CAR is involved. I think there's a whole other side of how you deliver and manufacture these cells. It's a big, a lot of the detractors to T cell therapy often point out how expensive and complicated the manufacturing process is, and how it's not really accessible to most people. And I think it's going to take a while. I think there's increasingly strides to make it cheaper and faster, and the manufacturing timelines keep getting shorter and shorter. But I think something I'm really excited about is the idea of going in vivo. Obviously, there was the acquisition of Capstan, the mRNA CAR company, which is exciting. That's one avenue. Also, there's lots of companies that are now thinking about delivering the virus directly to the human, right? Making a virus or a VLP or some sort of way of modifying the T cells, instead of ex vivo, do it actually in the blood of the patient. Those are actually working quite well. It seems like, at least to start. We'll see. They're not obviously in clinical trials yet, but it seems like that's an option. Um, I guess I get really excited about this idea that as we get better at engineering these cells, and we make this process cheaper and safer, and bring the costs down, and it becomes a more viable thing for lots of other diseases, this could increasingly be, in a similar way to mRNA delivery, a way that you deliver therapeutics in general for all kinds of diseases, is you deliver nucleic acids. Whether they integrate like a car would into the genome, or you transiently express an mRNA, just delivering things with new genetic code delivered directly to your cells, I think is such an exciting and powerful way to think about how to manipulate the body and manage disease and cure people of all kinds of things. So I'm just really excited about that. And obviously screening has a huge role to play, because figuring out what nucleic acid sequences to deliver is a huge problem, right? It's we need to learn this language of genetics, and we've obviously come a very, very long way, but it's so complex that screening and these machine learning tools are really going to help us get there.
So as a last little bit here, you did this work in grad school at MIT, at the Broad, with microbiome, and a lot of tools there specifically, and then you went and did immunology and then combined the two. Mm-hmm. So being someone who lives in both, what did you use from microbiome space that's really enabled you to do this work in T cells?
Interesting. I think a lot of the sort of the way you design the libraries and screen and think about the problems and the scale, I think comes in, even going back to the first CRISPR screens. I mean, people don't actually don't know this, or many people don't know this, but Feng Zhang, one of the inventors of CRISPR, was actually in George's lab and was around for a lot of these initial E. coli genome screening projects that we did with these like MAGE and these sort of like genome scale editing. And I think that probably informed a lot of the CRISPR screening revolution, and building libraries of guides and screening them. So I think this idea of doing things at genome scale really came from, as you say, from the sort of E. coli genomics folks like George's lab back in the day. I think thinking about the statistics and the sort of high throughput methods and the pooled screens, obviously that similarly was started in bacteria. And so I think a lot of those methods and statistical approaches and the way we clone things and think about the DNA jockeying of building these libraries, that also comes from that tradition, or that aspect of the biology. But as far as the thinking about the molecular biology of bacteria, it's pretty different. So I think that was one challenge for me, coming from a mostly computational and entirely bacterial PhD into immunology and T cells, and working for the first time day in and day out in the TC hood, and thinking about the timelines. The biggest thing is the timelines are so different, and how you plan out experiments. It's just like a rocket launch. It's like a 2-month long ordeal that you have to plan every single step, or everything goes wrong, you're done. Whereas I had the luxury in E. coli, if the experiment failed, I could just grow more cells tomorrow and it was fine.
Yeah. Eukaryotic cells are nice, but they're more demanding than E. coli, aren't they?
Especially, but T cells, I think, are the fastest dividing at their peak, 6 hours per cell division. I think that's the fastest dividing cell in the body. So I picked the right cell type, coming from bacteria.
Yeah, I mean, to be honest, every time I think of, like, for me, I thought I did all my training on T cell immunology, but they are exceptional cells when it comes to genome editing, gene editing in general. You can do so much with them, and they will take a lot of work.
They're easy to freeze and thaw, easy to expand.
Right? Almost if they want to be, I think that they just want to be edited. That is they are prepared for it.
And they're so easy to get from the blood, right? I mean, there's this whole aspect to it. Maybe this is also related to microbiome. You can take lots of people's T cells, and we have lots of clinical samples from healthy donors, and we can see what are the sort of genetic differences at the population scale, and how do those genetic and epigenetic differences among patients and different humans affect the T cell's function. That's also very easy to do in a way that it wouldn't be easy to do with a biopsy of another type of tissue. So I think the fact that they're so easily accessible and quite easy to get from peripheral blood is also quite a powerful thing to have.
Yeah, for sure. All right, so we're reaching the end of our conversation here. It's been super interesting. I could talk about T-cell engineering for hours, but we do have a limited time. And we want to ask you our fun question of the day in order to get to our listeners to know you a little bit better. And so we want to ask you if there is a hobby you've always wanted to pursue, but haven't had the chance to. Maybe you didn't have the time, maybe you didn't have know the place, or is there something in your to-do list that you'd like to get to?
Sure. Yeah. So I think, there's this cliche that maybe has mostly been debunked by now, but that, scientists don't do sort of art and humanities and that kind of stuff. And I think, there's so many counterexamples that maybe it's not true anymore, but I have always had a strong interest in music and art, and have tried to keep that up, mostly failed over the years. But one thing that, and I kind of am realizing this now, there's so many, well-known famous immunologists that are into music and play instruments. And there's, of course, the, what do they call themselves? The checkpoints, right? I spent some time hanging out with Jim Allison, and he would talk about playing with Willie Nelson and all that. So that got me thinking. Back in the day, I did a lot of music. I've come from a very musical family. My sister and brother are both musicians, and I was in the marching band and all these nerdy things. And I've always hoped and dreamed that I would come back to it. And maybe someday I'll go back to one of these immunology conferences and I'll play some jazz with some of my immunology idols.
That sounds like a great idea. If you are coming to IUIS, there will be a band playing there of fellow immunologists.
Cool.
But that is always so fun to see,
Mm-hmm.
especially in the conferences when there's some musically talented people take out their instruments. It's always super fun.
Yeah.
But I can imagine finding the time. It's pretty hard, isn't it?
Yeah. I think, I have 2 little kids right now, so that's my only.
Yeah. Which instruments do you play?
I play so mostly trumpet. That's the one that I spent the most time playing, and I love playing jazz on my trumpet. And I mentioned my kids, so guitar is a little bit more kid friendly. So we've been playing a lot of guitar together, and I've been teaching them chords and
That's
things.
what I'm doing. So my son and I take guitar lessons together.
Nice.
That's very nice. That's very nice. Well, then on that high note, pun intended, thank you so much for joining us. We had Dr. Daniel Goodman from the University of Pennsylvania, and the show today was a great conversation. Thank you so much for joining.
I love the conversation. I appreciate the time.
Thank you for being here. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. We're also on the socials at X at @ImmunoPodcast, Blue Sky at @immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.